This is a production of Cornell University.
Okay, so I'm going to be talking about malting barley today, which is barley that's used to make beer and whiskey, which makes for some fun phenotyping. Okay, so as many of you know, there's a lot of craft brewing that goes on in New York, so much so that we're the fifth largest producer of beer in the nation. Some of that growth has been spurred by the Farm Brewery Bill, which was passed by the state government in 2012. This gives economic incentives to local brewers to use local ingredients, ingredients being malt and hops. So right now, these farm brewers have to use 20% of their ingredients being from New York. However, by 2019, that's going to bump up to 60%. So as you can see, we're not growing a whole lot of barley in New York right now. It's creeping up, but we have some progress to make so we can meet that 90% minimum by 2024. So the Small Grains Group has been trialing malting barley and starting some research since about 2013. But for many reasons, which I'll go into, we decided to actually start some breeding research, which we're calling the Born, Bred, and Brewed in New York project, which I will hereafter refer to as the BBB. Okay, so our main goals here are to actually breed barley that's adapted to the unique conditions of New York, understand how genotype, environment, and G/E interactions affect malt quality across the state, establish best management practices for New York growers. Since this is a very new crop in New York, we still don't really know what the best way to grow barley is. And then variety trialing and recommendation. My work focuses on these first 2 points, which I'll be covering today. Okay, so before I continue, I wanted to give a brief overview of the malting and brewing process for those who aren't familiar. So with any crop, it starts with seed. I'm working in spring barley, so we plant this in mid to end of April and harvest in early August. So this is a picture of my nursery this summer at the Helfer Field. So you need clean, plump, disease-free grain to malt. Malting is 3 steps. You steep the grain so it imbibes water and can germinate. Germination is the second process. And then you dry it and kill at heat to develop enzymes, color, and flavor. So this is germinating barley right here. These little rootlets are called chit. And then this is an almost finished malt product. You can see the dried rootlets right here. This has been dried. So once you have the malt, you go on to the brewing process, which starts with grinding the malt, steeping it in water to remove the sugars. That gives you a kind of a malt soup, which is called wort. This is wort here. You add hops to the wort, you boil it to extract hop compounds and sterilize it, cool it, add yeast, fermentation commences, and then after fermentation, you sometimes filter it, let it rest for a little bit, and then you bottle it so you can enjoy it. Okay, so I mentioned that there are some challenges for growing barley in New York, mostly because of the weather we have. So this graphic is from the USDA, harvested barley acreage in 2016. As you can see, New York is producing so much barley that they didn't even survey New York. Most of it is being grown in the upper Great Plains and the Mountain West, where they have warm days, cool nights, and low humidity. Low humidity means low disease, and it also means that it will not pre-harvest sprout. So that's what happens when the grain has low dormancy, it gets wet before harvest, and the grain starts the germination process. This is bad because it means the barley does not malt very well, because malting is germination. The other big problem we have in humid areas is Fusarium head blight, which is a fungal pathogen that infects the developing spike and produces a nasty mycotoxin called deoxynivalenol. So even up here in middle of nowhere, Montana, malting barley is a tricky crop to grow. So the traits that we're really interested in and that are going to be key to actually getting malting barley to work in New York are disease resistance, pre-harvest sprouting resistance, and good enough malt quality that craft maltsters and brewers actually want to use this stuff. So most of our germplasm is actually from this area because this is where the breeding programs are. And then also Western Canada, they produce a very large amount of barley up there, but also European varieties from France and Germany. Germany grows a lot of barley, and if they can grow malting barley, I think that we can too in New York. Okay, so the method that we're using to actually go about achieving this goal is multiparental genomic selection in a reduced nested association mapping panel design. So I'll talk about that next couple slides. It's kind of a mouthful. So there have been a lot of genomic prediction studies within biparental families. And you can get pretty good prediction accuracies within biparental families for the most part. However, there are not that many in connected biparental families. These are two examples from Albrecht Melkinger's group in Germany. These are both in maize. So based on, from these publications, there is some promise that combining half-sib families can give you better prediction accuracies than just single biparental families. This is also a useful breeding model for us because there's one variety called AAC Synergy that we really like. It's from Western Canada, but its biggest problem is that it sprouts like crazy because it doesn't rain in Canada up there and they don't need sprout resistance. So we really just want to modify, we want to improve one trait, maybe two traits. And this, a crossing the single parent to seven other parents, excuse me, gives us a reasonable breeding strategy as well as academically interesting genomic prediction to look at. Okay. So the last year and a half to 2 years, I've been developing the strain population. I have about 1,400 two-row spring barley lines from 7 full sib families. Like I said, are connected by a single common female parent. I have 2 locations this year, and since I didn't have that much seed, I had to grow it in hedgerows in augmented design. Hedgerows are okay here because I'm not interested in yield right now. More interested in quality traits and disease, which I can phenotype in hedgerows just fine. I did a lot of phenotyping this summer. I'm actually still doing some phenotyping in the lab. And we harvested a subset of this to send to New Zealand for winter nursery seed increase so we can grow some of these in replicated yield trials next year. Don't know if I'll get to go there, but maybe next year. Okay, I actually don't have genotypic data yet. That's taken a little longer than expected. I actually just got the last 2 files back this morning. So if I were presenting in 2 weeks, I would have had some interesting genomic things to talk about, but you're stuck with phenotypic information, which is still kind of interesting too. So because this is an augmented design, oops, sorry. The entries are random, so I can calculate BLUPs, and they're nested within check. The heritabilities are kind of interesting here. So all that means I did all of the families at once. This is all 1,342 lines. And these are heritabilities within each of my 7 families. So you'll see there's quite a range across each of the traits: height, heading date, leaf rust, spot blotch, pre-harvest sprouting. Most notably, and then the genetic variants over here, there's quite a range, especially for height. And then some other traits too. One of the more interesting things you might know is that this SN family here tends to have lower heritabilities and lower genetic variants compared to the other families. This is actually a back cross. This parent is called Newdale, and it's one of the parents of Synergy. We didn't realize this until after the fact, but it was a happy accident. Should make for some interesting experimental things. This will not be being advanced in the breeding lines, though. Okay, and then I fit these with AR1 row column covariance structures in ASRMR. And the spatial effects did improve the model, so I kept them in, even though there wasn't noticeable spatial variation. Okay, so again, I haven't actually done a prediction yet, but I have a lot of interesting things that I can look at. I'll just start with our BLUP and probably Bayes CPI models, since I'm expecting for some of these traits there to be some major QTL segregating, major effect QTL. So the most obvious, I can start, finish up real well. Within family predictions, across family predictions, whole set to whole set. We also have a population from the University of Minnesota, 2-row spring barley. It's genotyped and phenotyped, so that's unrelated. Then I can do various combinations of all these. So I have a lot of work ahead of me. Again, I was hoping to present on some of this today, but come back next year and there'll be some good stuff. I also have kind of a mini experiment within the genomic selection experiment going, an early selection versus random, interested in looking at spatial correction versus just raw data. Because of the population structure and design, I should be able to run GWAS for this. We'll see if I have any major effect QTL that can fit as fixed effects. And because of the size here, I can empirically look at optimal size for the training set and how many markers. Okay, I need to speed through this. So I'm actually going to be doing selection too. So I'll be removing 2 families, crossing between all the others. phenotypic and genomic selection advancing simultaneously. There's not a lot out there in actually using selection indices for genomic selection. There's an Elliott Hefner paper from back in the day that just looked at it from a prediction standpoint, but didn't actually do any selections with the selection index. So because we're actually trying to do some breeding here, and I have a lot of phenotypes, I would like to look at that. Quickly, I want to talk about another project too that's related to malting. So malt quality is a super important phenotype. It's made up of 8 to 10 quantitative traits, so it's loads of fun. But it's really expensive, takes a lot of grain, usually in later generations, like any quality trait. There are micro malting machines, but they're also expensive, so I decided to make my own. This is a kind of a crappy cartoon showing a hydroponic ebb and flow table. So water moves up into this top tray. Barley grain is suspended in mesh bags. I have 10 grams, so I'm calling it nano malting. So I can fit a whole lot in this 3x3 table. The steeping takes place in here. The germination takes place in here. And this is all in a growth chamber to keep relative humidity and temperature constant. So I've done a couple test runs. These are my mesh bags here, and this is the grain. You can see the size here. I'm killing these in a food dehydrator. The phenotypes I'm interested in looking at are grain protein, malt beta glucan, and malt extract. And we have some nice regional trial grain samples from the past 3 years, winter and spring barley, multiple locations. So do a little G by E malt study, which would be fun. Okay, quick acknowledgements. Thank my committee, Mark, Gary, and Jessica. Everybody in the Sorrells lab, particularly James for lab and greenhouse help, David Bencher, Amy Fox, and Jesse Chavez for field help, David especially for doing all the disease inoculation, Jamie Cummings in the Bergstrom lab for making an inoculum, particularly the bipolaris, which is a big amount of work, so very grateful to her and the Bergstrom lab for that. Alyssa Blachez for pathology questions, Julie Hansen for letting me use the Vines lab NIR machine, Nick Kazmar for letting me use his ebb and flow table, which I will get back to at some point. It is a loan. And then Aaron McLeod from Hartwood College, who runs the Craft Food and Beverage Center and is a malting science wizard and has helped us out a lot. So yeah, think NY, drink NY, and I'll be happy to take any questions. It's here.
For older varieties from the UK, where they've got a long history of brewing, even more.
Yeah, so we've tested some UK spring lines. I think they might actually grow a little bit more winter grain because their winters are not nearly as harsh as they are here. But the UK material, somewhat surprisingly, is not done well here. We have, by far have the best luck in European germplasm with French and German varieties. And there are many French and German private companies that are actively involved in spring barley breeding, and Finnish too, but that's a pretty different environment from New York. So we're mostly focusing on like KWS, Secobra, Syngenta, brine material. Ed?
Cost effective at all to make hybrid barley varieties, and then if you do that,
yeah,
then you could do tricks on.
So there is hybrid barley in Europe, but it's primarily for feed. I don't know that anybody's looked at hybrid barley for malting quality. I think that you'd probably run into the same problem that hybrid rice has, that it might yield like crazy, but people don't like the quality. And malting barley the breeding programs are fully dictated by what the brewers want. And because of that, the North American 2-row barley germplasm base is extremely narrow because selection for the malting quality parameters for industrial brewing is so precise. There's not a lot of wiggle room. That'd be something interesting to look into, but I'm not aware of any hybrid malting varieties that are being researched. Jean-Luc.
For your nano malting, can you compare it to any full-scale malting or micro?
Yeah, so I should be able to compare it to micro malts. So the benefit of using those regional trials is that we send one rep of one location to the USDA Small Grains Quality Lab in Madison, Wisconsin. So I have a comparison there. I can also send some samples to Aaron McLeod. He has a micro malter. So, and I need to do that because I'm not going to be confident that I'm doing it right unless I have something to compare it to. Michael.
You mentioned
Yes. Yes.
So isn't a lot of the variation for the pre-harvest pro?
Yeah. So yeah. Yeah, well, so that one back crust that I talked about, that's also, they're both adapted to what they call the Canadian quality. So Canadian quality means that it yields well and it sprouts when you spit on it, because the big Canadian maltsters want to be fast and efficient. Um, there's also disease. I guess I was a little too simplistic in saying there's one trait we want to improve upon. We could improve some disease resistance as well. Um, I think so. It appears that the pre-harvest sprouting from the past QTL mapping papers in barley is oligogenic. It's not quite quantitative. And based on the heritabilities I'm seeing and my phenotypic distribution, I think that it's probably gonna be oligogenic. So I'm hoping to have some success in getting good prediction accuracies and actually selecting for pre-harvest sprouting, because we want it as low as possible. Yes.
You mentioned that there are hundreds of microbreweries in New York State. What are the differences among them? What are they varying in distinct?
That is a good question. So there is a skill level difference. You have guys that have been brewing in their basement for the last 5 years and decided to start selling it. And you have people that are professionally trained and they're doing this for 30 years. So there's a skill level difference. I would say that raw materials are probably pretty similar at this point. They all have access to the same malt. They could be buying New York malt if they wanted to, or they could be buying it from a large scale maltster in Oregon or Washington or Minnesota. Water can actually make a big difference in the taste of the beer and the quality of the beer. So if there are water differences across the state, that could potentially be part of it. But a lot of it's actually skill and the way you market it. And there are a lot of different beer styles that I could give a whole nother 30-minute seminar on. So maybe I could do that for a grass group sometime. People want to know about beer, I could do that.
One last question. If not, let's thank Dan.
All right, thanks.
This has been a production of Cornell University, on the web at cornell.edu.